Hello, my name is Ryoichi Matsuda. As Ugo told you that I retired University of Tokyo 2 years ago, and now I'm working in a private university named Tokyo University of Science. It's also in Tokyo. And also I'm serving the chairman of International Biology Olympiad to encourage high school students who want to be life scientists, biologists, or doctors. And the participating country is over 75 now, and soon will reach to the 80, including Italy team. But anyway, today I'd like to talk about two topics. One is dietary PI phosphate overload. That's very bad for muscle. So if you prevent the intake of excess amount of PI phosphorus, that'd be good for your muscle, is what I want to say to you today. And also the second topic is vitamin C. This is like antioxidant, like Dr. Hayashi mentioned minutes ago. Vitamin C is the same story, but it's very good for muscle and prevent muscle degeneration, at least in culture. So I will tell you about this work. Let me explain the dystrophin because I am concentrating to work on the Duchenne type muscular dystrophy. That's missing dystrophin in the skeletal and cardiac muscle of the patient. Okay, the dystrophin was discovered 1987 by Lu Kunkal in the States. And dystrophin is, as you know, that it's a rod-shaped protein. It connects to cytoskeleton and cell membrane. So, and the dystrophin resides underneath the cell membrane of muscle fiber. So this is healthy muscle from normal person. And this is a section from DMD patient and stained with anti-dystrophin. You cannot see any positive signal. Okay, so the dystrophin, the function of dystrophin is like working as a rivet of this airplane structure. So if you have a frame here and if you have a outer wall, it's like a cell membrane. The rivet connect between this frame and outer wall. So if you don't have rivet enough, your airplane will have a big problem. Like in 1985 in Japan, the Japan airline Jumbo jet crashed in the middle of the mountain in Japan and killed 520 people. It was the biggest single airplane crash in the history. And this was caused by the mistake of riveting. So somebody during the repair process, the engineer forgot to put rivet at the right number at the right place. So during the flight, because of the so huge stress, made this frame and wall detach. And this made big air leakage in the cabin and blew up the wing, and the airplane couldn't be controlled by pilot and crashed into the mountain. So it's terrible. And if you have a right number of a rivet at the right position, this framework will work, but without rivet will make a big problem. And in the DMD, Duchenne type muscular dystrophy patient, it looks like the rivet is missing, or the number of rivet is missing in their muscle. That's a problem. And after that, if the muscle contract, cell membrane start to break and make a leakage from the outside, like from outside, the calcium or the serum protein will get into the muscle cell, and also the muscle protein will leak out from this damaged membrane hole. So like the famous story that the creatine phosphokinase comes out from muscle cell to the bloodstream, makes creatine kinase in the serum is a good marker for the this membrane condition in the patient. So the CK in the blood is high, much higher than normal. That means you have a problem on the in the muscle membrane. And the same thing, the calcium will get into cytoplasm. And also if you have a vital staining dye, such as advanced blue, which is my favorite dye, will go into the muscle, inside of the muscle. So if you have a calcium in the living cells, cells start to contract. This is hypercontracted muscle fiber. So the advanced blue is here, this reddish fluorescence is advanced blue. Okay, so this muscle fiber tend to round up. So this means muscle fiber is contracting, so hypercontracting. So we call this opaque fiber. So by using this advanced blue vital staining dye, you can actually differentiate the degenerating muscle fiber and, sorry. Degenerating muscle fiber and muscle fiber without any membrane damage. But eventually these muscle fiber will start to follow this sequence and make muscle degeneration. So using this method, you can actually count the number of degenerating muscle fibers. So you can use this method as a evaluative for the evaluative purpose of your gene transfer or muscle cell transfer or stem cell transfer or chemotherapy. Using this method, you can quantitatively evaluate the therapeutic effect. So this is again muscle. This is a advanced blue incorporated muscle. That means a degenerating fiber here. This is a breast muscle. Also, some fiber is healthy, but other fibers are stained with blue. That means you have they have a membrane damaged muscle cell here. But to get the advanced blue staining image, you have to sacrifice the MDX mouse. It's a mouse model for DMD. So you have to sacrifice them. But using X-ray micro CT without sacrificing, still you can see the muscle degeneration. But in this case, we found some white spot appeared in the degenerating muscle fiber, degenerating area. So if you make a section and stained with Koser, this stains calcium. So you can see the calcium positive deposit exists in that advanced blue positive area. So if we use electron probe microanalysis in the degenerating area, you see no signal on the carbon, but strong signal in the phosphorus or calcium. So this deposit might contain calcium phosphate. And if you check the signal using energy dispersive X-ray spectroscopy, you can find out that the deposit is a hydroxyapatite. Okay, and this calcium positive area, alizarin lead positive area, corresponds to the area of macrophage positive area. So the calcium depositing area or area causing ectopic calcification corresponds to the inflammatory response. And if you check the concentration of calcium or phosphate, phosphorus, or the creatine kinase activity in the serum from B10, this is the control, and this is MDX mouse. Actually, you see higher level of phosphate in the MDX mouse. Calcium is almost the same in the serum, but as I mentioned, because MDX has many muscle fibers with membrane leakage, so the serum contains a very, very high level of creatine phosphokinase activity. So their serum contains higher level of phosphate than the control. So if we check the concentration of each phosphate or in the control or MDX and different young and a little bit older animals, you will see like this. As they getting older, the phosphate phosphorus concentration increases. And in human case, human patient with ectopic calcification also show the higher level of phosphate or phosphorus. So our conclusion is MDX mouse contains ectopic calcification in their skeletal muscle and also their cardiac and kidney also. And the main component is hydroxyapatite, and the incidence of ectopic calcification increases with age. Inflammatory macrophages accumulate in the calcified area, and increased phosphate concentration is apparent in MDX serum. So we wanted to see the effect of phosphate, inorganic phosphate in mouse diet. And if we let them to eat for 2 months, and we see the change, whether or not they change the skeletal muscle pathology. Okay. This is a hematocrit L-zinc staining of a control B10 mouse and MDX mouse. You see something occurring in this area. So contains many nuclei. This must be the measure of inflammation or muscle degeneration. And this is MDX mouse, so no dystrophin. And if you stain with Evans blue, you see many Evans blue positive fibers only in MDX skeletal muscle. And alizarin red is here. It shows calcium deposition. And if you keep the mice, this is a B10 mice with a high phosphate diet, and this is MDX mouse with low phosphate diet. This is MDX mouse with a normal concentration phosphate diet, and this is a high phosphate concentration diet, and ate for 2 months. And if we stain the calcium in the transplanted leg muscle, you will see many positive. This is a calcium staining. So many ectopic calcification occurs in the MDX mouse skeletal muscle cell fed with high phosphate diet. But if you reduce the level of phosphate to like 70% of the normal diet, you will see the big difference in terms of ectopic calcification in skeletal muscle. There are some, but very low compared with MDX mouse fed with normal diet or MDX mouse fed with a high phosphate diet. And even the fed with high phosphate diet, beaten control mouse, wild type mouse, they don't develop such ectopic calcification. So the ectopic calcification is very interesting and may have some molecular mechanism which might explain the muscle degeneration in dystrophic muscle. Here, it's a 3D X-ray CT image of the MDX mouse fed high phosphate diet for 2 months. So you see the many ectopic calcification deposit here. And if you keep 1 more month, keep them for 1 more month with low phosphate diet, the deposit density decreases. Oops. So this means ectopic calcification is reversible. So you can actually rescue them using low phosphate diet. Okay, but if you keep them eating for 2 more months, for 1 more month with high phosphate diet, look here, you see so many calcium deposit here. So it's very bad for muscle. And if you check the inflammatory factor response using RT-PCR, MDX mouse fed with low phosphate, normal level phosphate or high level phosphate diet, you will see the inflammation marker such as interleukin-6 or tumor necrotic factor alpha or interleukin-17 or RUNC increase. And this is internal control of the GAP-DH. So this means if you have a high level of phosphate in your diet, muscle of the MDX mouse start to build up the inflammatory response. And if you reduce the level of phosphate, you can actually rescue them. And this is we check how they can run using a voluntary wheel running low phosphate diet. The mice they are eager to run, but the high phosphate diet, the mice they don't move so actively. And if you check the histology, high phosphate diet, the mice express many regenerating fiber, contains embryonic myosin heavy chain, and also macrophage marker and the necrotic pathology. So only the PI uptake make big difference in the MDX mouse skeletal muscle. So it's good news for them. So our conclusion too is phosphate or phosphorus might regulate muscle degeneration. And if you lower the phosphate level, you can actually rescue at least MDX mouse. We like to try this to the human, but since I'm only the mouse doctor, MD, but not a medical doctor, so I cannot treat the patient with this kind of strategy. So some of you in the audience, if you're a medical doctor and can treat the patient, I wish that you can try this experiment using human patient, maybe less phosphate only. That makes probably a big difference. And if we check the muscle power in the low phosphate diet treated mouse or high phosphate, the low phosphate MDX mouse, they have a much stronger power generation. So again, the phosphate level in diet is important. And if you can reduce the level of phosphate using just a normal diet, that'd be good for patient. So we used calcium-based phosphate binders, which binds to phosphate and reduce the uptake of phosphate into the bloodstream. They reduce the phosphate from the food in digestive tract, I think. So using this Renagel and fed with normal diet, okay, Renagel, the inflammatory response reduced and necrotic area also reduced. So the Renagel or phosphate binders might be way to go to rescue the patient. And this Renagel is applicable for patient with kidney disease. They have some trouble to excrete excess level of phosphate. So this is can be used for patient. So if you use Renagel for DMD patient, might have some better beneficial effect is our guess. It's the same thing for Renagel. Same thing for Renagel. So Renagel is a very good, can rescue muscular dystrophic phenotype, at least in MDX mouse. So our conclusion 3 is here. You can read them from the recorded video. So let me move on, since the time is limited. Or let me explain about our vitamin C story. As you know that the primates, humans or monkeys, we have gene, it's already a destructive gene, broken gene. It turns out a pseudo gene for the vitamin C synthetic enzyme. So primate needs vitamin C from outside, but dogs, mice, cat, they can produce their own vitamin C using healthy gene for vitamin C producing enzyme. Okay, so vitamin C deficiency occurs mostly only in the primate. And vitamin C, ascorbic acid, tend to degrade so fast if you add ascorbic acid into the calcium media. But its stable vitamin C exists. It's L-ascorbic acid 2 phosphate. So this one can stay for several days at a very high level compared with plain ascorbic acid. So using this ascorbic acid 2 phosphate, we can actually check the effect of vitamin C in cultured skeletal muscle cells. This is cells came from mouse brain tumor. It's called BC3H1. This is very interesting cell because they express myogenin and myosin heavy chain. So, but no cell fusion. So people say that this is defective cell for muscle differentiation defective cells. But if you add vitamin C, ascorbic acid 2 phosphate in the culture media, you see myogenesis and myosin heavy chain expression and nice collagen formation and the myogenin expression also. But if you have a collagen inhibitors, cis hydroxyproline, the myogenin expression disappeared, and no muscle fusion, no myosin heavy chain expression in this collagen inhibitor containing culture media. Okay, so myogenin upregulation caused by ascorbic acid 2 phosphate is somehow related to collagen synthesis. And as you know that the transforming growth factor beta is also inhibitory effect for myogenesis in culture and in vivo too. But if you have a vitamin C in the culture media in presence of transforming growth factor beta. Vitamin C can overcome the inhibitory effect of transforming growth factor beta. So this is a bad condition for muscle cell. They don't make a nice muscle compared with control. But if you have a vitamin C in presence of a TGF-beta, they can make vitamin C as almost as a control. It's good news for them.
3 minutes left.
Okay, okay. As you know that the TGF-beta accumulate to aged muscle and sometimes makes aged muscle atrophy. So what I'm thinking is I'd like to give the ascorbic acid to the aged people. Same thing for low temperature treated muscle. At the low temperature muscle cell don't differentiate, but in presence of vitamin C, they differentiate. Yeah, let me explain about the mitochondrial membrane story too. If you have a vitamin C, this is activated mitochondria stained with this red color, JC1. In the myotube, they have activated mitochondria. But at the low temperature, no mitochondrial activation. But if you have a vitamin C, this case in presence of co-presence of IGF-1, but without IGF-1, still the vitamin C can activate membrane potential of mitochondria and also stimulate myogenic expression. So this is a phosphate. If you have an excess amount of phosphate,
it is pity, Ryoichi, it is pity that you do not left 2 minutes for discussion.
Okay, okay. Oh, I see. In presence of a high level of phosphate, muscle cells don't differentiate. But in presence of vitamin C, also they can differentiate. So, so vitamin C works for TGF-beta treated or high phosphate treated or low temperature treated. So there are many ways to rescue muscle, differentiation, inhibition condition. But we like to see the effect of these things in living animal. So this is what we are doing now. And then eventually by vitamin C or Renagel or other stuff, we like to rescue the muscular dystrophy patient. And these are the name of my co-worker working in the University of Tokyo or Tokyo University of Science. Okay, thank you very much for listening. And I'm happy to take your question in 10 seconds.
Since we are 2 minutes in advance, there is time for 2 short questions and very short replies. No one? 1, 2, 3, we move to the next speaker, which is Hai Wu Hong. Is this correct? Hai Wu Hong is here.
Yes, I'm here. Thank you for inviting me.
You are here. Yes. Your microphone is on. I will cancel my microphone. Thank you very much, Ryoichi. But next time, talk less and discuss more.
But there was still some 1 minute to go, right? I mean,
yes, 1 minute.
It was a beautiful presentation.
Yes. It was wonderful, but too much.
The story about the phosphorus and the vitamin C. So bottom line is phosphorus is bad and ascorbic acid is very good. Is that correct?
Yes,
correct
yes.
Story, right?
Yes.
Several lines of evidence showing that phosphorus is bad.
So if you can reduce the level of phosphate, that's good news for muscle, dystrophic muscle, to keep them healthy.
Okay, thank you very much.
Second question from myogenesis people. No question. Next year. We go to Hong Hai Wu. Thank you very much, Professor Matsuda.